Hello, everyone. Can you hear me? Perfect. I was trying to fetch two of our other presenters from the lounge. Sorry for the delay. I will get started in 1 minute. So, hello, everyone. Welcome to the STEM Student Talk Session 4. I have an announcement to all of the speakers and presenters in the room. Can you all please request share audio/video so you're in the queue for me to accept you once it's your turn to present. Also, please have your slides ready in the tab to share screen. Perfect. So let's get started ahead. Hello, I'm happy to welcome everyone to the STEM Student Talk Session 4. I'm Claire from Polygence, who will be the moderator for this session. We have a full roster of amazing projects to share with you, and we have 4 presenters covering a variety of topics. And I want you to all know that this is a safe and friendly environment. So presenters, you don't have to feel nervous. Please feel comfortable and just be excited to share your passion today. So in the room today with us is Alex Gwinnett, who will be the judge of our session. He's currently training in the UCLA Caltech Medical Sciences Training Program, and he completed 2 years from MD from UCLA, and he's currently studying as a PhD student at the Caltech Institute. Alex will be scoring each student's presentation along with myself. And each presenter will get 8 minutes to present. And when the presenter has 2 minutes remaining for their talk time, I will silently rejoin the screen so that the speaker will be aware of the time. During presentations, I would kindly ask the audience to type your question in the chat for each speaker and make sure that you put your question in the Q&A tab under sessions. We will allocate 2 minutes for questions after each presentation. So first up is Audrey from Houston, Texas. Second, we have Julie from Nashville, Tennessee. Third, we have Aditya from Frisco, Texas. Last but not least, we have Shirzad from Westfield, New Jersey. So if Audrey, you're ready, I will invite you up to the podium and let's get started with your presentation.
Can everyone hear me? Okay, so hello, my name is Audrey Ajakaye, and I am a high school junior from Houston, Texas, and today I will be presenting Mind the Skin podcast series. So, as I mentioned before, I'm from Houston, Texas, and I am the CEO and founder of my own company called Love My Skin, which this podcast is a branch of. So, why Mind the Skin podcast? So, my podcasts are between 8 to 10 minutes, and they aim to find the connection between mental health and skin conditions. This field is known as psychodermatology. So, why did I decide to do this? So, this is because many teens struggle with skin conditions like acne, eczema, and excessive sweating, and these can be difficult to treat and can really take a toll on one's mental health. So, I wanted to find out scientifically what are the reasonings behind this? And so currently I have 3 episodes and they are entitled Acne and Stress, Acne and Excessive Sweating, and Eczema and Anxiety. So I decided to do this, as I mentioned before, because the skin and mind are intricately intertwined, and this can be explained through neuroscience. So these are the 3 episodes that I have. So if you would like to create a podcast, I want to take you through some steps that you can use to have a successful one in a simple and easy way. So first, you want to start with marketing and technical research. Next, it is vital to have a Polygence mentor that can help you. Third, you want to have a script that you write. Next, you want to plan out how the podcast is going to be structured. And next, you want to have equipment, software, and music to get your podcast edited and started. After that, you will record and edit your podcast. And finally, you will host and grow the podcast. So first, I'm going to start with market research. So the Mind of the Skin podcast series was really started for three main reasons: people, passion, and personal interest. So first, I wanted to engage people in a less formal way on how to better be educated about how their mind and their skin can be connected. Next, I have a true passion for this area, as this is what I plan to study in the future. And third, I figured that creating a podcast would be a good way to get all people of all ages to have good personal interest in this. And so first, for my market research, I started with creating a word cloud, which is where I sent a survey to 150 kids that were in my grade and asked them, when they think about their mind and their skin, what are the main words that come to mind? And so these, which you see on the screen here, were the ones that were the most popular. And so these are the words and the topics that I use to frame my podcast episodes. So next, I had the help of a Polygence mentor. So finding a Polygence mentor was crucial in developing my podcast because my mentor, Dr. Deborah Oyeyemi, was an expert in the field of neuroscience, and she gave me a lot of technical advice, which I used when writing my script and especially with research. There are many qualified Polygence mentors that you can use out there as well for any topic that you choose to do your podcast in as well. So next was the script and editing plan. So for me, I leveraged whatever background I had. And for me, that was really my background in STEM from my school. And so from then, I started with a broad outline of topics and went from there. So that included analyzing articles, medical journals, research papers, and like stories or testimonials. And within this research, I was really focusing on the topics of mental health and skin conditions and insecurities. And I discovered that there's actually a field that connects these two called psychodermatology. And so I was then from there able to condense everything into technical research into key paragraphs, which I used to frame my script. Next, I edited my script, and my mentor, Dr. Deborah Oyeyemi, helped me to refine some things and fact check a lot of the information I gave. And so one big thing that I want to share with you guys is that there is no right or wrong way to write a script. As long as you stay true to your vision and stay true to your passions, your podcast will be amazing. So next was equipment and software, recording, and editing. So for my podcast production, I wanted to have a cost-effective and simple way of going about it. So I only use three main things. I use my script, I use my iPhone to record, and I use iMovie to edit. And so the bottom line, what I want to share with you guys is that you don't need to have fancy microphones or fancy editing software to edit a podcast, but you can do it really with what you have already laying around. So the final thing is when you want to host, publish, and grow your podcast. So there are many ways to distribute and market your podcast. And there are some streaming services like Podbean and Buzzsprout that allow you to simultaneously stream your podcast to the major podcasting services, which are Spotify, Apple Music, Amazon Music, Pandora, and many more. So this is one sneak peek of my Mind the Skin episode that I would like to share with you guys now.
Hello everyone. I'm Ajakaye, and this is Mind the Skin, a podcast where we break the boundaries between neuroscience and dermatology to answer questions with our audience in mind. I'm a high school senior interested in the connection between the mind and the skin. I've always been passionate about skin health, and I've seen firsthand how painful and distressing skin conditions such as acne and eczema can be for younger and older people. Do you know that skin conditions can result in just as much disability as other chronic diseases, affecting self-esteem, school, work, and even relationships? You've been listening to Mind the Skin podcast, the number one stop for everyone who wants a better understanding between the mind and skin connection. Thank you everyone for listening to my first podcast. Have you ever wondered how eczema and anxiety are connected? Well, join me next time on Mind the Skin podcast. Until then, so long and be safe.
So that was just a sneak peek of my first episode, which is about acne and stress. And I will be releasing that very soon onto all the major podcasting services. And so finally, I just want to thank my mentor, Dr. Deborah Oyeyemi, who was able to really guide me throughout this process. So like I mentioned before, my podcast will be on all major streaming services coming soon. And if you have any questions, you can contact me via email or Instagram. Thank you so much for listening.
Amazing job, Audrey. Thank you so much. I will now remove you to the podium. If you guys have any questions to Audrey, please type it in the chat. Right now, Julie is trying to fix her audio problem. So next, I will invite Aditya for the presentation.
Yeah, hello. Give me a second to screen share. Yeah, hello everyone. So you guys can all hear me, right? Okay. So my presentation is on LRRK2 and its importance in Parkinson's. Just a little bit more about me. I'm from Frisco, Texas. I love everything about neuroscience. So this is why I decided that I'm going to look a little bit more into one of the most common brain diseases we know. So there's just a little bit of a structure before I start my presentation. I'll have 5 main sections. First, I'll just give you an overview of like what Parkinson's is and some of the common symptoms. And then I'll talk about LRRK2, like what it is and why it's important. We'll go a little bit further, and I'll just warn you right now. Number 3 will be a bit more technical. I'll just give you a jargon warning. So if you have any questions, just leave them for me, and I'll explain any terms that in more depth later on in the Q&A time. After that, I'll talk about some possible treatments, which is really the meat and why LRRK2 is so important, and I'll close everything up with a conclusion. So to start us off, Parkinson's is a really common mental disorder. It usually happens in more senior citizens, but there are cases of Parkinson's appearing in people who are younger. Although it's mostly balanced, there is a little bit more cases towards the male side than the female. And for location, so Parkinson's is really unique in that you find it in the neurons of a certain region of the brain. It's a very small region. It's called the substantia nigra. And for some reason, scientists have not figured this part out yet, but for some reason, it only affects those cells, even though LRRK2 is found everywhere because it's a protein. It's found in all cells, but it only seems to have an effect in that area of the brain. So Parkinson's is split into 3 main causes. environmental, sporadic, and genetic. And I've given like a basic description of each of them here. Now, people generally like to combine environmental and sporadic because that unknown cause element is sort of a commonality between them. But there are sporadic genetic causes, so I wanted to like make sure to make the distinction here. So just a little bit further of a look into where the substantia nigra is. As you can see, it's a really small region, and it's only those two lines that you can see on the left and right hand side of the diagram there. Those dark and black lines, that's where the substantia nigra is, and that's when those, if you look at the bottom portion there, they die off under Parkinson's. So what is LRRK2 and why is it important? LRRK2 it's been proven to be the most common genetic cause of PD. Remember how I split the three causes, Parkinson's, into three different causes? So of the genetics category, LRRK2 is the most prevalent. Although that number may seem low, 5 to 13%, it's a lot because of how common Parkinson's is. And if there's any familial or just genetic causes, whether it's mutation or passed down from a family member, there's a chance it can be from LRRK2. So the major thing that causes Parkinson's LRRK2 is mutations, and LRRK2 is a protein, so it has a gene that codes for it. So when that gene gets mutated, that's why Parkinson's happens. So the function of LRRK2, it's part of a cell signaling pathway, and this pathway, it regulates cell function, specifically membrane trafficking, which is basically when a cell creates molecules and exports them out of the cell, or within parts of it, that's called membrane trafficking. So when it is disrupted by a mutation or whatever, that can cause a problem, which is how Parkinson's develops. So what changes does a mutation in LRRK2 do? All the mutations that we found, or that scientists have found in LRRK2, cause it to be overexpressed. That just means that too much of it has been made. And when there's too much of the protein, it can cause a lot of problems, which is why it has a big effect. So this diagram on the right here, it's just an image that has been recently been developed of what LRRK2 looks like, its protein form. And what LRRK2 does is, as I said, it's a membrane trafficking, part of membrane trafficking signaling pathway, but it involves proteins from the ROBGTPase family. Now, this family is very important because what it does is it's a family of proteins that has various important jobs of regulating things inside the cell. So LRRK2 is like the managerial protein that tells these what to do. So when it gets overexpressed, or there's too much of it produced, that means that it keeps activating these ROBGTPases over and over again more than they should be, which is that's the root of the problem. So this is kind of an example of like what LRRK2 does. So if you look at the diagram on the right, you can see the process of dopamine being secreted in these dopamine producing neurons. Now, in this chain, if there's a problem, or if LRRK2 gets overexpressed, this dopamine can't be released properly, and that's what causes these neurons to die or to cause problems, because this isn't being properly done. Now, why is this so important? The most important part of LRRK2 is that it has because it's such a big scope, right? Because it has so many cases. If we can figure out how to solve LRRK2, we can figure out how to solve a lot of affected patients. So an example of like current drugs that are happening is this L-Dopa. If you go back, if you can look in this, you'll see next to number 2, it says L-Dopa. L-Dopa is a precursor to dopamine. So when you ingest L-Dopa, you basically are artificially taking dopamine. And this is a good symptom reliever. It will help you get through some of the worst effects of Parkinson's, but it will wear off after time because as the body recognizes foreign dopamine, it will start to reject it. So this is an example of something that cures symptoms but won't work full time, and that's why LRRK2 is so important. LRRK2 is special for two really big reasons. It's a central figure in a lot of these cellular pathways. It's basically what I would call a hub, in which case if you change it, it will change everything that it's connected to, which means that instead of having to take 5 different medications for 5 different symptoms, if you treat LRRK2, you can treat all those symptoms at once, which is a really big thing. And the second thing is that I was talking about sporadic cases, right? And those are cases where it's unknown. And there's been some proof where LRRK2 has shown potential to help treat patients whose cause is unknown, which makes it super important because if it's mass produced, it can change a lot of people's lives. So the biggest thing that I would like people to take away is that although it's potential, keep in mind nothing is confirmed yet, looking at LRRK2 and looking at cures for LRRK2 or using it as a cure will be amazing for, especially if you can treat unknown causes for Parkinson's. So right now, LRRK2 is undergoing a lot of research in labs and by drug developers. But what I want people to see here is that if you know anyone, or if you ever happen to get Parkinson's, I would consider looking into clinical trials, because this will help not only help everybody get the treatment faster, but it will there's a possibility that you'll be able to get a drug that will treat it faster than you might have otherwise. So these are just my sources, and I'm good for questions.
Great job, Aditya. Such a mind-blowing study. And now I will remove you from the podium and have Shirzad up next to present.
I think it should work now. Can you guys hear me now? Okay, all right. Let me screen share. Okay, so hold on. All right. Hi, my name is Shirzad Mustafa. My mentor was Tariq and Dale, and today we'll be talking about my project, and it's on manga and anime and tools to improve mental health literacy. This this project is about how manga and anime can be used as a tool to help people understand the broad term that is mental health. And yes, first I would like to share a video summarizing a good portion, or that serves as a very good introduction. So yeah, I'll play it right now. Okay, that's a bit strange. As a matter of fact, it's okay. I'll still continue. So manga is a literary form of entertainment rooted in rich Japanese art with spiritual, social, and philosophical messages. It's a Japanese comic art that has also evolved into animations or anime, which is extremely popular among teens. Um, uh, uh, the crisis at hand. Mood disorders and suicide rates have increased over the last decades among adolescents and young adults. The COVID-19 pandemic has only compounded the problem, and that can be seen a lot. Manga in education and health promotion. Yoshika Okuyama manga's depiction of characters as differently abled and influenced Japan's cultural constructions of shogai, which means disabilities, and promotes social change. Manga is used to educate people on this topic and prevent bullying, and also manga is used to improve physical activity, promote healthy eating, and reduce disease-related stigma over the years. Okay. So the general understanding for mental health literacy is that the knowledge of mental disorders, risks and causes, self-treatments, professional help available, and attitudes that promote recognition and appropriate help. And for those of you who have more questions about this, feel free to ask them. I would love to answer all these questions if you have any. So purpose of the current study. There are not a lot of work related to using manga and anime for MHL, which is mental health literacy. The study aims to investigate adolescents' perceptions of manga and anime as a tool for improving mental health literacy, which is extremely important to me. The hypothesis is that participants will perceive manga and anime as a contributor to their increased mental health knowledge, empathy, and identification, and most importantly, hope-seeking behavior. Hold on. So design, procedure, and measure. So in this study, there were 75 participants recruited through the researchers of my TikTok channel with roughly 320,000 followers and directed to my Discord server and Twitch to access the link to self-administrated survey. This survey contains 23 questions. The survey contained 23 questions, included demographic data, participants' exposure to certain anime and manga, whether reading or watching any manga with mental health content made the participants more knowledgeable and aware of mental health and generated empathy of those with mental illness. As with the results, 57.5% of the participants were male, 34.2% were female, and 8.2% identified as non-binary. The participants ranged in age from 15 to 25 years old. 39.4% were and then 35.2% were 16 to 18 years old, 12.7% were 19 to 20, and then finally 12.7% were 21 and 25 years old. Results continued. So the data goes a lot more. The data is a lot more general. So I'll list very specific statistics. 40.5% of participants were familiar with Evangelion, and it slowly decreases as the manga and anime continue on. 66.7% of the participants identified depression as the most talked about mental illness in the manga and anime they have read or watched, followed by anxiety with a 59.4% and suicide and ideation 58%. And finally, 73.6% of the participants indicated that manga or anime made them think more about these mental health issues. So since this is general information, general graphs, I will list more statistics. 89.6% of the participants from this study indicated empathy for the manga or anime characters with mental health illness. 86.6% of the participants indicated empathy for those they see around them who have mental health issues due to manga or anime. Finally, 83.3% of participants indicated that they would seek help for themselves or others. 75.8% of the participants indicated that manga and anime made them understand the importance of seeking help from mental health professionals. Okay. So interpretations and limitations. The results indicate that mental health focused manga and anime can increase mental illness knowledge and understanding and promote help-seeking behavior. However, there were a lot of limitations. For example, recruitment was voluntary from any manga channels specifically and self-reported and not followed over time. Possible use. The research can help facilitate the use of manga and anime as a format for mental health outreach and education. Multi-component school-based intervention, community-based social service intervention to reach hard to reach and hard to serve adolescents and teens. Peer training to promote emotional intelligence and empathy. So recommendations and further studies. Randomized controlled trials, systematic reviews, and meta-analysis are necessary to demonstrate the efficacy and effectiveness of manga and anime. Studies from scientific disciplines, including brain research and natural sciences, to explain the mechanisms underlying the effectiveness of manga and anime. Exploring the psychological and behavioral theories, the social cognitive of modeling characters engaging in health behavior. Narrative or storytelling approach to increase accessibility, and graphic effect of enabling sensory understanding. Acknowledgements. So I would like to thank everyone who stayed around and listened to me. I would also thank my mentor who helped me a lot, and I appreciate all of his help. And his name is Tariq Endale, and I appreciate his guidance. Stacy Hill, Charlotte Wetwerb, Brianna Chrisman, and the Polygence team for their support and for giving me this wonderful opportunity. And Olivia Rose for actually reading thoroughly through my very, very rough draft and giving me all the advice I need. And yeah.
Perfect. Great job, Shirzad. Last but not least, we have Julie. So Julie, take it away.
Okay, so I know I had audio problems before, but can you hear me now? Okay, that's great. Okay. And can you see my screen as well? Okay, so my name is Julie Key, and I'm a senior from Nashville, Tennessee. And during my Polygence project, I researched the topic of epigenetics. This project culminated in a scientific paper, which I plan to submit to a journal for publication very soon. And one of the main goals of my paper was to explain epigenetics in a way that compares it to traditional genetics, which is something that people might be slightly more familiar with. And my research was also focused on the ways that studying epigenetics with different techniques can be applied to the medical field as well, particularly diagnostics, which is something that I'll discuss later in my presentation. So first, I want to give some background knowledge about my topic. Epigenetics is the study of how external factors from the environment can affect how our genes are expressed. So unlike traditional genetics, epigenetic mechanisms don't induce disease by changing the order of the DNA bases themselves. Instead, they'll alter gene expression levels, which basically means it'll affect the amount of proteins made. Therefore, disruptions to epigenetic activity is implicated in a multitude of disorders, including cardiovascular and autoimmune diseases, neurological disabilities, and cancer. One example of an epigenetic mechanism that is important to my presentation is DNA methylation, which involves methyl groups, which have a chemical formula of CH3, and they're binding to DNA. This will be important later on when we discuss the utility of measuring DNA methylation levels in specific genes as an indicator of cancer. Okay, so here on the screen, I have a figure that I actually made for my research paper, which places epigenetics in the context of the central dogma of biology, basically meaning that DNA is transcribed into mRNA, which is then used to make proteins. And to illustrate the relationship between epigenetics and disease development, I want you guys to consider a comparison between epigenetic diseases and monogenic disorders. So monogenic disorders are more straightforward in the fact that they're caused by a single mutation to a single gene. These mutations can lead to abnormal changes in the molecules that make up proteins called amino acids. And this ultimately results in errors to protein structure and an overall defective or misfolded protein, as you can see in part B of my figure. But on the other hand, epigenetic diseases affect gene expression without changing the DNA sequence itself. And this means that it actually influences the degree to which genes are transcribed, and therefore, instead of resulting in protein misfolding, epigenetic mechanisms like DNA methylation commonly involve changes to the amount of protein made, and this ultimately results in more complexity with epigenetic diseases as opposed to monogenic ones, since variation in the amount of protein produced can actually correspond to different levels of the disease's severity. And something that's interesting about epigenetic mechanisms is that they can actually have physical impacts on the three-dimensional chromatin structure. And so chromatin is basically just a substance that is composed of DNA strands and the proteins that the DNA is wrapped around. And most of the current scientific literature on how epigenetic mechanisms control gene expression supports the hypothesis that chromatin condenses and decondenses to regulate transcription. So typically, when chromatin is in its compacted form, it's considered transcriptionally inactive, meaning that the DNA sequence on that region of chromatin will not be expressed. And conversely, when DNA is uncondensed, it's considered transcriptionally active because the looser structure of the chromatin will open it up to binding by proteins like transcription factors, which are needed to express gene function. So taking DNA methylation as an example, the biochemical change of adding methyl groups to chromatin results in its physical compaction and prevents transcription. And due to these functional effects, identifying when and where these changes occur can serve as a diagnostic marker for disease. And here I want to use DNA methylation again as a model to demonstrate the diagnostic applications of studying epigenetics, specifically in the context of cancer. So oftentimes in cancer cells, methylation will occur at genes responsible for maintaining genomic stability, thus inactivating the transcription of genes capable of really important functions for your body, like growth suppression and DNA repair. This is the case with the adenomatous polyposis coli, or APC gene, which does help to suppress tumors. And when this tumor suppressor function is disrupted by DNA methylation, it can contribute to sporadic colorectal cancer. In fact, what I have here is that one study found that hypermethylated APC promoters were present in 44% of breast tumors and 53% of non-small cell lung cancer tumors, showing a really strong correlation between methylation and malignant cancer development. In contrast, only 4% of non-cancerous tissues showed methylation of the APC promoter. And this is just one example of dozens of promising epigenetic biomarkers that I encountered during my research. And thus, if scientists are able to pinpoint the methylation levels of disease-relevant genes in patients, it can serve as a powerful indicator of early-stage cancers. And now that I got kind of a better understanding of the relationship between epigenetics and disease, I wanted to see what methods were currently available for quantifying epigenetics. And the most common methods I found were sequencing-based techniques. And while sequencing was conventionally used to determine the order of DNA bases, more recently it's been co-opted to study epigenetic mechanisms as well. So sequencing technology is responsible for much of the information that we currently have on epigenetic mechanisms in specific tissues, since it's capable of identifying like methylation levels in DNA, and it can sequence the presence of proteins to infer whether regions of chromatin are considered open and active or closed and inactive. This can be used to identify specific epigenetic biomarkers, like with the APC promoter that I mentioned in the previous slide. However, unfortunately, preparing cells for sequencing does often involve fragmentation and extraction of DNA strands. And thus, one of the main limitations of sequencing is that it can't show chromatin structure and its folding processes in a spatial context. However, a technique able to do just that is a relatively new field of super resolution microscopy or imaging, which is actually one of the ideas that I do propose in my paper for future directions of study. Using microscopy techniques helps to retain spatial information of chromatin without disrupting its natural structure. So what this means is it's basically really important for examining the interactions between biochemical modifications like DNA methylation and physical features of chromatin, like the level of its compaction. And it also lets researchers kind of get a better insight into what these characteristics look like in diseased cell contexts. And as microscopy continues to make progress in cost effectiveness, resolution, and efficiency, it can become a powerful tool for understanding and diagnosing epigenetic diseases in patients. With that, I also want to give a huge thank you to the Polygence team, as well as my mentor, Alina, for guiding me through the scientific literature review process, as well as research and writing my own paper. Thank you for listening, and I hope you learned something about epigenetics today.
Great job, Julie. I'm so glad we fixed your audio issue. So thank you so much to all of the presenters today for all your hard work. And also a huge thank you to all audience members for coming to support these students. We had an amazing variety of topics in this room, including psychodermatology, Parkinson's disease, anime and manga, and epigenetics. We at Polygence are incredibly proud of all of your achievements, and we're so glad that we got to cheer you on and celebrate you on the stage today. So we would like to encourage you to attend and support other students' presentations throughout the rest of the day. And you can just simply navigate back to the reception area and click on other sessions. So that concludes this session. And in a couple of minutes, we will have a wonderful talk from our keynote speaker, Margot Gerritsen. She's a Stanford data science professor and a co-organizer of the Global Women in Data Science Conference. So please join us on that stage, and you can do so also by simply navigating back to the reception area and clicking on the keynote speaker stage. So thank you, guys.